everyone. Hope you all are doing good. Welcome back on my YouTube channel. And today we are starting the chapter 5, that is fundamental unit of life. So what is the meaning of fundamental? Fundamental means basic or important. So what is the basic or the important unit of life that we are going to study in this chapter? As you know that any building, when building is constructed, it is made up of bricks. So what is the basic unit of a building? That is a brick. In the same way of any living being, the basic unit is a cell, because any living being, whether the animals, the plants, the microscopic organism, these are made up of cell. So cell is the fundamental unit of life. Now students, the question is, who discovered cell? Cell was discovered by Robert Hooke. He was a scientist who discovered cell in 1665 by the self-designed microscope. Actually, he was examining cork. Now the question is, what is cork? Cork is a substance which is made by the bark of the tree. And students, if you have seen the old trees, the big trees, the stem of these big trees have bark on their stem. If you observe the stem of the old tree, you'll find that some old wood type structure and such kind of structure is there on the stem of the plant or the tree, and that structure is basically bark, and from this bark the cork is made. Cork is a substance which you can see sometimes on the bottles. Nowadays even rubber cork are coming in market. You can see in the lab also rubber cork. Earlier these cork were made by the wood. It is very soft wood, and that was taken from this bark of the tree. So when he, while examining this cork, the slice of cork, basically what did he see? He do? He just took a thin slice, and from that thin slice, he observed this thin slice under the microscope, and then he saw such kind of structure which resembled to the honeycomb. Bees honeycomb, you can see this is the structure in this. He just compared this structure with this honeycomb, and then he gave this structure the name cell, because he compared it like the small rooms are there, many small rooms he compared like in this way, and then he gave the name cell. Microscope which was designed by Robert Hooke is like this, and you use this microscope for watching the slides in your lab also. So what is important to learn in this microscope? You will learn the design also, what is the design and what are the parts, because the questions are asked on the basis of this. You can see this is the first part, that is the eyepiece. Sometimes the question comes, how many lenses are there? So there are two lenses in the microscope. One is the eyepiece and one is the objective lens, and one mirror is there. You can see this is the mirror. So what is the function of this mirror? The light which falls here on the mirror, it reflects. You can see it is reflecting here, and then this is the slide, and in this slide the object specimen is put, and this slide you can see this is just clamped with the help of two clips. You can see, and this board which is on which the slide is put, it is called stage, right? And these are the coarse adjustment and the finest adjustment. With the help of these adjustment we can adjust the distance between the slide and the lens to make the picture clear. We can just adjust with the help of these coarse adjustment and the fine adjustment. This part is known as the arm of the microscope, and this part is known as the body tube of the microscope. So this all is about the parts of the microscope and how many lenses are used in a microscope. There are two lenses used and one mirror is used. Now students, see how to prepare a temporary mount of onion peel. This is the practical, and because of COVID you are not going to the lab. So here I am showing you how to do this practical, and you have to learn these all steps very carefully, because sometimes you will see this question may be asked in question paper, that some steps are given and you will be asked that arrange these steps in a proper sequence. So how is it done? First of all, you will take an onion, and this onion you will just cut as it is, like in 1/4 piece you can cut. And you know that there are so many layers of these fleshy scale leaves are there in an onion. So you will take one of that leaf, and then from this concave side you will just take out its peel, and it is very easy. You can snap the leaf backward so that you can see the epidermis layer, and then you will take a thin layer of epidermis, and with the help of this forceps, you can see this is the forceps, and with this forceps you can just peel it out, right? This is just membrane, you can see when you will cut this onion. And then this is the slide, this square glass, this rectangle glass is the slide, and on this you will just pour a drop of water, and on this drop of water you will put this cut piece of epidermis or the onion peel. Why is needed the water to keep this? Because otherwise this membrane can be folded. So to avoid being folded, you have to put water here, and then you will take this cover slip and you will just put this cover slip and you will press the cover slip here. Then you will pour a drop of stain here on one end of the slide, and then this stain will just come inside also, and it will this piece will be stained or will be colored. Why stain is necessary? Because if you will not stain the slide, the proper structure you will not be able to see. So that is why this stain is also compulsory, or and this stain which you will use here, that can be safranin or iodine, right? And then you can just a small piece of filter paper you can take, and you can extra stain you can wipe it off, and then you can observe this slide under the microscope. While observing under the microscope, you will find such kind of structure. In this you can see not all the organelles visible here, only the nucleus is visible and cytoplasm is visible, and this is the cell wall which you can see. So this simple structure can be observed under this simple microscope. Now see, these are some discoveries about the cell, and this is again important to learn. As you know that Robert Hooke discovered cell. He noticed the presence of cell in the Koch's lice in 1665. Then Leeuwenhoek in 1674 he found the presence of the living cells in the pond water. Robert Hooke in 1831 recognized the existence of the nucleus in the cell. So who discovered nucleus? Robert Brown. Then Purkinje invented the term protoplasm, which is liquid present in a cell. Means the cytoplasm and nucleoplasm together, he coined the term protoplasm. Then Schildin and Schwann in 1838-1839 they presented the cell theory that all organisms are actually made up of cells. And then Virchow also in 1855 suggested that all cells come from the cells that already exist in nature. So what was the cell theory given by Schwann and Schildin? The first point, that a cell is the structural and functional unit of all living organisms. So why is it called the structural and functional unit of all living organisms? Because all living organisms are made up of cells. So cells give structure to the organisms, as well as cells are essential for performing various life processes required for sustaining life. So that is why cell is the structural and functional unit of a living organism. Next is all the living organisms are made up of cells, and cells are formed from pre-existing cells. If any organism you see, it it just comes from the pre-existing cell. A single cell divide and multiply and then form an organism. So from the pre-existing cells, the cells are formed. And organisms are of two types: unicellular organisms as well as multicellular organisms. Unicellular organism means that they consist only one cell, single cell, like amoeba, and multicellular organisms which contain various cells that perform different functions in the organisms, such as the plants, fungi, and other animals. So you can see here that there are different type of cells. This is the red blood cell, this is muscle cell, nerve cell, then paramecium, this is diatom, and this is the leaf pore of the guard cells. These are the guard cells, this is the structure of stomata. So you can see that these all cells are different in their shape and sizes. This is oval in shape, then on the other side this muscle cell is pointed on both the sides. So why do they have the different shapes? And if you see nerve cell, this is again peculiar in shape. This is paramecium, slipper in shape, you can see. So why do these all cells have the different shape and sizes? In order to perform the specific functions, because each cell has to perform some specific function. So that is why they have the different shape and sizes. And each living cell has the capacity to perform certain basic functions. That are the characteristics of all living organisms, and each such cell has got certain specific components within it known as cell organelles, and these cell organelles together constitute the basic unit that is called cell. I hope you all have understood whatever is discussed in this video. So that's all for today, and thank you for watching the video. We'll meet in the next video again. Thank you. Bye bye.